Hello everyone, welcome to the session. So in this session we are going to discuss about the CUET PG exam. Okay, so in this session I assure you I can give you a complete analysis of the question pattern as well as the syllabus analysis. Okay, so this video is specifically for the zoology syllabus. Okay, so the first thing will be you should know what will be the basic understanding of the CUET exam. Okay, so the mode of an examination will be online, so you should know that this is a CBT test, that is computer based test and online. Okay, so it is the mode of examination will be online. The medium of the question paper will be English and Hindi. Okay, and the duration of the examination will be 2 hours. Okay, so that is 120 minutes. And the frequency of exams in a year, how many times an exam can be conducted in a year, that will be once in a year. Okay, yearly once the CUET exam will be conducted. So the types of question, it is an objective question, surely you get 4 options, so that is multiple choice questions, MCQs. The total number of questions will be 100. The total marks of the question paper will be 400. In the sense, you should know that one question carries 4 marks, so total marks will be the 400. And the negative mark, definitely there is a negative marking, so whichever the exam you are dealing you are having the negative marking, you should be very cautious with that. Why? Because so the negative marking, you can lose marks very easily. Okay, so here the negative mark is there. So the marking scheme will be, you can get 4 marks for a correct answer and -1 for the wrong answer. Say for an example, you will get the first question right and the second question is also right and the third question is also right and the fourth question is wrong. Okay, so for the 3 questions you will get 12 marks and the fourth questions you have attended this question and you have attended wrongly. You have chosen the answer wrongly. In the sense, you can get 12 minus 1, 11 marks. So this is called as, this is how the negative mark has been calculated. Okay, so the total sections in the total number of sections in the question paper will be 2 sections. Okay, section A and section B. Okay, so what section A carries? So the section A consists of language comprehension, verbal ability or general knowledge questions, mathematical ability and analytical skills. So these are all the pattern of the section A. And what is with respect to the section B? So this is our major subjects. Okay, domain specific questions. For example, our zoology questions will be coming in the section B, right? So now, what are all the units coming under the zoology? So they, in the official website, they just give the title of the units, each units. For example, unit 1 cell biology, unit 2 biochemistry, unit 3 molecular biology, unit 4 ecology, unit 5 evolution, unit 6 biodiversity and environment conservation, unit 7 immunology, and unit 8 genetics, unit 9 biotechnics, unit 10 vertebrates and invertebrates, unit 11 developmental biology, unit 12 biostatistics, unit 13 animal physiology, unit 14 recombinant DNA technology. So totally we are having 14 units. So in this 14 units, they just given the title, okay, for each and every unit. So based on the PYQ analysis, I have come into a conclusion for in which unit, which is subtopic the questions has been frequent. Okay, so now the first unit, that is cell biology. So in the cell biology, the very important subtopics have been prokaryotic and eukaryotic cells. You should know the basic difference between prokaryotic and eukaryotic cells. Okay, and the cell theory and the cell size. Who proposed the cell theory and what the cell theory is there. For what is the cell theory is explaining, and the plasma membrane structure and function. So this is very important. Transport across membrane, that is diffusion, osmosis, active transport. So all these things will be coming under the plasma membrane only. So cell junction, so we are having 3 different types of junctions: tight junctions, adhering junctions and gap junctions. Okay, so we have to understand the basic ideology of these three junctions and the organelles. So the important organelles present in the cell will be the nucleus, mitochondria, endoplasmic reticulum, Golgi complex and lysosome. So these are all the cell organelles. This is not only the organelles present in the cell, but there are also other important organelles is also there, but here where the questions is very frequent. Okay, so cytoskeleton, microtubules and microfilaments. So this is the very important cytoskeleton. There we're going to study what are all the what is mean by this microtubules and microfilaments and what their function in cell cycle and cell division. Okay, so the next important thing will be the cell cycle, and in cell cycle you should know we are having a G1 phase, S phase, G2 phase, M phase and G0 phase. We are going to analyze top to down in regarding with all these cell cycle as well as the cell division. So in cell division we are having meiotic division as well as the mitotic division. Mitotic division is takes place in which cells, meiotic division is taking place in which cells. We are going to analyze everything detailedly in our classes. Okay, so now, based on the cell biology chapter, you should know in which topic the questions are frequent. Okay, so in the previous year questions, in the questions have been arised from the fluid mosaic model. Okay, so they have given some four statements and you have to identify the correct statement. Okay, so the questions are frequent from the fluid mosaic model. That is nothing but this is the model with respect to this plasma membrane. And the second one will be the cellular organelle functions of mitochondrial inner membrane. Okay, so the cellular organelle mitochondria, the question is very frequent. And the ribosomes. We are having 70S ribosomes and 80S ribosomes, where this 70S ribosomes will be present and where this 80S ribosomes will be present. Everything will be discussed detailedly in our class. Okay. And another important cell organelle, the lysosomes, which is also called as the suicidal blacks. The question is very frequent. And what is the main function of this lysosomes? So this is acting as the suicidal blacks. Why we are calling this lysosome as a suicidal bugs? Because this lysosome have a millions of hydrolytic enzymes where the pH will be acidic in nature. So that pH is suitable for the hydrolytic enzymes to survive in the lysosome organelle. Now the cell cycle phases, the question has been arising frequent in the cell cycle phases, that is G1 phase, S phase, G2 phase and M phase. Okay, and what is the difference between mitosis and meiosis? So you should know the basic details between this mitosis and the meiosis. So this is where these are all the important topics where we can find the questions very frequent. Okay, so the which organelle is rich in cardiolipin? So for that you should know the basic understanding of each and every organelle. Okay, what it is exactly composed of, and which phase occupies maximum duration, which means so in this G1 phase, ES phase, G2 phase and M phase, which phases occupies a maximum duration. So you should know in 24 hours cell cycle has been done. One cell cycle has been completed in 24 hours. So in complete cell cycle will be occurred in the 24 hours. We are having different phases in each cell cycle. So among all these phases, which phase occupies the maximum duration? This can be expected in the question. That's all about the first unit. So the second unit will be the biochemistry. So in the biochemistry, what are all the things you have to study under this chapter biochemistry? That is biomolecules. So in the biomolecules, we can study about this carbohydrates, lipids, proteins and nucleic acid. So these are all the basic biomolecules you have to study. Okay, so the enzymes, so in enzymes we are having different types of enzymes: lyase, ligase, oxonuclease, nucleases, lipases. What are all the types of nucleases? What are all the properties of enzyme? So this enzyme is changing what is exactly the enzymes, what it is do with respect to the rate of reactions. So that is what we are going to study in the properties of enzymes. Now factors affecting activity. What are all the factors which is affecting the activity of the enzymes? So this is very important. Now, vitamins and coenzymes. What is the relation between these vitamins and coenzymes? First of all, what is mean by coenzymes? Everything will be discussed detailed in our classes. And metabolism basis. So here where we have studied the carbohydrate chemistry, lipid chemistry and protein chemistry. So here we are going to study about the carbohydrate metabolism, lipid metabolism, protein metabolism and nucleic acid metabolism. Okay. So metabolism in the sense that has been either it will be catabolism or anabolism. Catabolism in the sense many complex molecules. Complex molecules will be breaking down into a simpler molecules. So in the anabolism, many simpler molecules together they are forming the complex molecules. So this is what a catabolism and anabolism. Together we are calling them as a metabolism. So that is what glycolysis. This is a carbohydrate metabolism, Krebs cycle and electron transport chain. Everything will be discussed detail in the metabolism process. And beside all these things, we have to study what is pH, what is the basic understanding of this pH, buffers and buffers in the biological system. For example, bicarbonate ions will be acting as bicarbonate. Bicarbonate is acting as a buffer in human blood system. So we are going to discuss detailedly in the classes. Okay, so based on the PYQs, what are all the questions which are very frequent in the examination? That is the level of protein structure. So in the protein structure, we are having primary structure, secondary structure, tertiary structure and quaternary structure. What is the structure of primary protein, secondary protein, tertiary protein and quaternary protein? For example, the primary protein, the structure will be linear. In the secondary and tertiary, in the quaternary, what will be their structure? In which structure the loop is going to form? For all these structure, which bond is necessary? Which bond is necessary to make all these structures? Everything will be discussed detail in our class. And enzyme specificity. So when it comes to the enzyme specificity, you should know two important models. One is lock and key model and another one is induced fit model. Okay. So in the lock and key model, you should one lock there is only one key. Okay, for one substrate there is only one enzyme. So this is what for this in this substrate only one enzyme is going to associated with this substrate. So this is what a lock and key model, for one lock one key. Okay, for example, in the induced fit model, based on the substrate structure, the enzyme is changing their structure. By changing the structure, it is going to fit with the substrate. Okay, so this is what a difference between the lock and key as well as the induced fit model. We are going to discuss detail and complete analysis with respect to this enzyme specificity in our classes. And competitive and non-competitive inhibition. So we are having an inhibition. So the one substrate is going to, one enzyme is going to bind with another substrate. They are going to give an enzyme substrate complex. As a result, they are giving a product and the enzyme will be dissociated from the substrate. This is how the properties of an enzyme. But how we can inhibit the enzyme? So based on the inhibition, we are having two different types, that is competitive inhibition and non-competitive inhibition. Okay, so in that competitive inhibition and non-competitive inhibition, we are having a Michaelis-Menten equation. So in the Michaelis-Menten equation, we are having Km, Vmax. So what is Km and what is Vmax? Everything will be discussed detailedly when we see about this Michaelis-Menten equation. Okay. So next, the question will be frequent in the glycolysis process. Okay, where exactly this glycolysis is going to take place, and in the process of glycolysis, how many net number of ATP we are going to yield. Okay. So these are all the area we can see the frequent questions. Now, which bond stabilizes an alpha helix? As I said before, so in the secondary structure, we are having alpha helix and beta pleated sheet. So which bond is necessary for alpha helix? Which bond is necessary for beta pleated? As these bonds are stronger, they can make the secondary structure, tertiary structure and quaternary structure. So which inhibitor increases the Km but not Vmax? So this is very important. So we are having a competitive inhibition, non-competitive inhibition. So in the competitive inhibition, Km is going to increases or Vmax is going to increases. So we have to study like this. By comparing all these things, we have to study. So that's all about the second unit, biochemistry. In the third unit, that is my molecular biology. So you have to understand what is the difference between this DNA and RNA. Okay. So what is the difference between DNA and RNA? It is just, you should not stop just by knowing the full form. That is deoxyribonucleic acid is DNA and ribonucleic acid is RNA. You should not stop up to that. Beyond all that thing, you have to know some more points. Okay. And after that, you should know what is replication and what are all the enzymes are involving in replication, and what is transcription, which enzyme is very important to play a crucial role in the transcription and translation. Okay. So DNA to DNA, we are going to call about that as a replication. DNA to RNA, we are calling them as a transcription. RNA is getting converted into protein. That process is called as a translation. So all these processes are coming under the unit called as a molecular biology. Okay. So about after that, so in the translation, protein synthesis is going to taking place. What is protein? Many amino acid together, we are calling them as a protein. Okay. So in that, what is genetic code? For each code, how the amino acid is going to link with that. Okay. So we are going to see all these things in the class. So where we can expect a question. Okay. So direction of DNA synthesis. You just keep a note of that. Always the DNA synthesis will be in the 5' to 3' direction. Okay. The parent strand will be always in 3' to 5' direction, and the synthesis of DNA will always be in the 5' to the 3' direction. Okay. So this is where we can expect a question, and many students will face difficulty in remembering these things also. And what are all the enzymes are involving in the replication? Okay, so the main important enzyme will be the DNA polymerase. In case of transcription, it is a RNA polymerase. In case of translation, it will be the polypeptidyl synthase. Okay, so these are all an important thing. And when it comes to an RNA in the transcription, not only the mRNA is going to produce. Okay, we are having three different types of RNA: mRNA, tRNA and rRNA, based on their function. In case of mRNA, it is just decoding the information, that is messenger RNA. Okay. So this tRNA, it is a transfer RNA involving in transferring the amino acids, and this is the ribosomal RNA involving in protein synthesis. Okay, based on the function, we have categorized RNA into three types. Now genetic code. Why we are calling this genetic code as a degeneracy and ambiguity, universality. All these words are very, very important. You should have a note of that. And what is this start codon and stop codon? We are having totally 64 codons. Okay. So in this 64 codons, 61 codons are the sense codon. Remaining 3 codons will be the nonsense codons. That is UAA, UAG and UGE. So these are all the stop codons. Whenever the translation process is in the translation process, if this codon is there, the translation process will be stopped, which means it will be terminated. So these are also called as a terminator codon. So then what will be the start codon? Always AUG. It codes for an amino acid called the methionine. So this is considered as a start codon. Okay. So in the previous year questions, what they have asked in a previous question, DNA polymerase adds nucleotides in which direction? As I said before, it will be in the 5' to the 3' direction. Which codon codes for the methionine? That is AUG. So these are all the basic questions. So based on the PYQ pattern, you can easily come into a conclusion. If you know the basic understanding of each and every topic, you can easily crack the examination. Now the fourth unit will be this ecology. So in this ecology, you should study about this level of ecology. In the sense, so a group of individuals, we are calling them as a population. Group of population, we are calling them as a community. Group of community, we are calling them as a ecosystem. Okay. So the ecosystem components and energy flow. So the ecosystem components, if it is an ecosystem, how can we define an ecosystem? An interaction between a biotic and abiotic factors is called as an ecosystem. So what is biotic factors? What is abiotic factors? If it is an interaction between these two, how that interaction will be? Everything will be studied in this ecosystem components. And energy flow. How the energy flow will be? The energy flow will be always unidirectional. Okay, it is not bidirectional, it is unidirectional. Now biomes. Okay, now population dynamics. In the population dynamics you should study about the growth curve. So growth curve, in the sense, whenever you are coming across this topic called growth curve, you should know what is exponential growth curve and what is logistic growth curve. Okay, so in the logistic growth curve you can find an important constant that is called as a carrying capacity, K. Okay, everything will be discussed detail. And what is succession, that is ecological succession, and what is ecological pyramids. Okay, so in the ecological succession we are having primary succession and secondary succession. Okay, what is primary and what is secondary? And in ecological pyramids we are having three pyramids, that is pyramid of energy, pyramid of numbers, and pyramid of biomass. So if you know in which pyramid whether it will be upright or inverted, that's all about this topic. Now conservation biology basics. Okay, so whether it is important to conserve our biodiversity or not. If it is important to conserve our biodiversity, what are all the basics of the biodiversity conservation? Okay, so whether it will be ex situ or in situ. What is meant by ex situ, what is meant by in situ? That's all about this chapter. So in the basic, in the PYQ analysis, they have been we can come across the questions from the topic called as the food chain and the food web, and the ecological pyramid based on the number, biomass, energy with respect to this aquatic ecosystem and terrestrial ecosystem. And what is mean by this 10% law? Okay, this 10% law is also called as the Lindeman's rule. Okay, so this population curve, that as I said before, exponential growth curve and the logistic growth curve. So exponential growth curve, in the sense we are having a J-shaped curve, and it is not realistic in nature in case of environment. And in with respect to this logistic curve, we are having a sigmoid shaped curve, S-shaped curve. And whenever we are saying explaining about this logistic curve, why we are having this S-shaped curve is because of this carrying capacity of that particular ecosystem. So in the PYQ we can expect a question, which pyramid is always upright, whether it will be pyramid of numbers or biomass or energy. Who proposed the 10-man's rule? As I said before, it is also called as a Lindeman's rule. So we can easily come up into a conclusion. So the fifth unit will be this evolution. So whenever we are studying about this evolution, we should not skip the scientist Darwin. Okay, Charles Darwin, and the synthetic theory, speciation mechanism, Hardy-Weinberg equilibrium. This is very important. Based on which criteria? Which 5 criteria Hardy-Weinberg have been proposed this Hardy-Weinberg equilibrium. Okay, so this adaptive radiation. What is adaptive radiation? What is convergent evolution? What is divergent evolution? See, from one common ancestor, they have been, it leads to speciation over a particular period of time. So this is called as a divergent evolution. This is with respect to this adaptive radiation. Okay, so what is the difference between this convergent evolution and divergent evolution? What is mean by analogous structures and homologous structures, and what is adaptive radiation? Everything will be discussed detail in our classes. So based on the PYQ analysis, the question will be frequent from the Darwinism, and what is the difference between Darwinism and Neo-Darwinism, and what is Hardy-Weinberg principle, and what are all the causes of deviation from the equilibrium, which means if the Darwin have been explained the 5 conditions through which the population will be the genetic drift will be constant. Okay, so that is what a principle of Hardy-Weinberg. So if there will be any deviation from these 5 factors, it should not be an equilibrium. So what are all the deviations? Okay, so as I said before, what is homologous organs and what is meant by analogous organs? The question, so say for an example, no migration. Okay, so if the population, if it is not leads to an evolution, say for an example, what is Hardy-Weinberg equilibrium? In the sense, this is an ecosystem. Okay, so in this 5 conditions, I'm going to say about what that 5 conditions, and that 5 conditions, Okay, so if this 5 conditions persist in this equation, this ecosystem will not face any evolution, will not face any mutation, and will not face any variations. Okay, so this is what an Hardy-Weinberg equilibrium. And what are all the 5 conditions? That is, no migration between in the species will be taking place. No migration, no mutation, okay, random mating, okay, larger population. Okay, so based on these criteria, he have been proposed the Hardy-Weinberg equilibrium. If the deviations from these 5 factors, then then the equilibrium will not be maintained in this particular ecosystem. So this is how the previous question will be, and the simplest question. Biodiversity and environmental conservation. So in the biodiversity, you should study about taxonomy and systematics. What is meant by taxonomy and what is systematics, and what are all the levels of biodiversity. So when it comes to the level of biodiversity, we are having 3 different types of levels. That is genetic biodiversity, species biodiversity, and ecosystem biodiversity. We have to study the levels of biodiversity and what are all the threats to biodiversity, and we can call this threats to biodiversity as an evil quartet, which means 4 important things which is causing a threat to biodiversity. And the first important thing will be the habitat loss and fragmentation. We are going to discuss that. And what is meant by endangered concepts, and what is this organization which is called as an IUCN, International Union for Conservation of Nature, and how can we conserve the biodiversity. And we are having 2 conservative strategies, in situ and ex situ. What is the difference between these two, and what are all the examples for this in situ and ex situ? So in the PYQs, we are having questions from the levels of biodiversity, endemic species. So what is mean by endemic species, in situ and ex situ? And what are all the hot spots? How many hot spots throughout the India? Okay, how many hot spots in India, and what are all the pots? Okay, so here the in the previous year questions, we can have a question called, which is an example of this ex-situ conservation. Okay, we have found this question. Now immunology. The 7th unit will be this immunology. So whenever you are studying, I will start to study about this immunology, you have to understand the basic difference between innate immunity and adaptive immunity. Okay, and what are all the cells which are involving in the immune system? Say for an example, T cells and B cells are involving in the immune system. And what is the difference between antigens and antibodies? So if there is any foreign particle enter into our body, we are calling them as an antigen. And if the antigen enter into our body produce an antibody which is fighting against this antigen, and that is called as an antibody. Okay, so humoral, what is the difference between humoral immunity and cell mediated immunity? Which cell is involving in humoral immunity and cell mediated immunity? We are going to study about that. And what is meant by vaccines? What is the basic ideology in the vaccines? Okay, and what is the relation between this vaccines and herd immunity? Everything will be discussed here. In the previous year question, what is this innate difference between this innate immunity and acquired immunity? We can expect a question from here. And what is the antibody structure, that is Fab region and FC region, that is constant region, antibody fragment of antigen binding site. Okay, so types of immunoglobulins, basically we are having 5 different types of immunoglobulins. Which immunoglobulin is also called as an antibody. Which antibody is involving in which functions? We are going to study about that. And how the antigen and antibody are specific with each other. Okay, so if the certain antigen enter into our body, it will be bind with this only with specific antibody. So that is what antigen and antibody specificity. Okay, so which immunoglobulin crosses the placenta? So even though the concepts are very critical, the questions are very basic. Okay, so we are having 5 different types of antibody: IgA, IgA, IgG, IgM, and IgD, and IgE. Okay, so these are all the 5 different types of antibodies. In these 5 different types of antibodies, IgG has a capacity to cross the placenta. Okay, so this is the basic question we can expect from the immunology chapter. So in the 8th chapter, that is genetics, we definitely in the genetics chapter we should not skip the Mendelian laws. Mendel have 3 laws: law of dominance, law of segregation, and law of independent assortment. And what is linkage and what is crossing over? So what is the difference between this linkage and crossing over and the Mendel's third law called the law of independent assortment? We are going to discuss about that. And what is chromosomal aberrations? We are having 4 different types of chromosomal aberrations, that is deletion, duplication, inversion, translocation. Okay, so these are all called as the chromosomal aberrations. Sex determination. So what is in sex determination, we have to study about male heterogamety and female heterogamety. Okay, we are going to study about this. And what is extra chromosomal inheritance patterns? So in the previous year, we can expect we have a question from the Mendelian ratios. For example, in the monohybrid and dihybrid cross, we are having phenotypic and genotypic ratio. So in the monohybrid cross, the phenotypic ratio will be 3:1 and the genotypic ratio will be 1:2:1. Okay. So this is then the dihybrid cross. In the dihybrid cross, the genotypic ratio will be 9:3:3:1. So this is how the cross ratios will be. Okay, we are going to study about that. And what about this incomplete dominance and codominance? Okay, so we are going to study about and which is codominant, the example of codominance and the examples of the incomplete dominance. Now, so we have to study about this linkage and crossing over types. Okay, so in the linkage we are having coupling linkage and repulsion linkage. And in the chromosomal aberrations, as I said before, in the deletion and inversion, the questions are predominant. Now, so in the PYQ, the crossing over, in the PYQ questions, we are having this crossing over occurs during which stage? Obviously, that will be in the Pachydian stage. Which ratio indicates the incomplete dominance? That will be 1:2:1. Okay, so how this 1:2:1 have arisen, everything will be discussed in the class. So biotechnique, in the biotechnics, so we are having a instrument, that is PCR, gel electrophoresis, chromatography, spectrophotometry, basics of molecular cloning methods. All these methods are very, very important. We should not skip these techniques. In the PYQ, we have the same instruments. We are expecting a question. Okay. So what is the principle of this polymerase chain reaction? What is the principle of this gel electrophoresis? How it is getting separated? So based on the charge, it is separating. Okay. So chromatography, what is the principle of chromatography? And spectrophotometer, it is based on which law? That is Beer-Lambert's law. So the question will be, the DNA fragments are separated in gel based on the charge. Okay, based on the charge they are separating. So the 10th unit, that is vertebrates and invertebrates, that is completely related to non-chordates and chordata. So right from the phylum Porifera, Cnidarians, Platyhelminthes, nematodes, arthropods, mollusca and echinodermata, they are coming under the invertebrates. Okay, so you should study about the general characteristics and classification of all these phylums as well as the examples. These are very important. If you study the basic general characters and classification with example, that's all about each and every phylum. Also, each phylum have unique characteristic features that cannot be seen in any other phylum. So you have to see about that. And chordate classification. So in the chordate classification, we are having protochordata to mammalia. So we have to study about what is protochordata and what is excuse me, what is vertebrata. So key adaptations and life history. So these are all very important. So these are all the questions we can expect. We have questions in the PYQ. Okay. So based on Porifera, Annelida and arthropods and larval forms. So trochophore larva and bipinnaria larva is the larval forms of which of the following phylums? And chordate features. What are all the general characteristic features of the chordates? And what are all the differences in these fishes, amphibians and reptilia? So these are all the class coming under the phylum chordates. Okay, so you should know the characteristic features of each and class coming under the phylum Chordata. So in the PYQ we have a question, which larva is seen in the echinodermata. Okay, so that's all about this 10th unit, and the 11th unit will be this developmental biology. So in the developmental biology, you should study about sperm gametogenesis. So in the gametogenesis, we are having two gametogenesis process. One is spermatogenesis and one other one is oogenesis. How the sperm is going to produce, how the egg is going to produce. So all these things we are going to study in the gametogenesis. How the sperm and the ovum is going to fuse with each other. That fusion is called as a fertilization. As a result of fertilization, we are going to get this zygote, and the zygote is undergoing a repeated mitotic division. That division is called as a cleavage pattern. As a result of this cleavage, we are having a formation of these three germinal layers. That formation of germinal layers is called as a gastrulation. In each and every germ layer, the different organs is produced, that is called as an organogenesis. And in this organs, which is the stem cells, from which stem cells which is going to produce, that is called as a stem cells. Okay, so the questions can be arising from the types of cleavage. What is holoblastic cleavage? What is meroblastic cleavage? So everything will be discussed. And gastrulation. So what is the three germinal layers? Okay, so ectoderm, endoderm and mesoderm. From the ectoderm, which organs are going to produce? From mesoderm, which organs is going to produce? From endoderm, which organs is going to produce? So all these things will be studied in the gastrulation process. And in the germinal layers, and in which layer which organ is going to derive. That is called as a derivatives. So this which germ layer forms a nervous system. So this is how the question will be. Obviously it will be in the ectoderm. So the 12th unit will be this biostatistics. So in the biostatistics, you should know what is meant by data. What are all the types of data? So how can we calculate in the form of mean, median and mode? Okay, so how we can calculate the standard deviation and variations. What is the probability basics and applications of statistics in biological research? How the statistics will be helpful in the biological research. So the questions will be from the mean, median and mode. How to calculate this mean, median and mode formula are very important. Standard deviation, graphical interpretations. Okay, so which measure is the least affected by the extreme value? So this biostatistics will be discussed detailedly in our class. So animal physiology. So in animal physiology, you should know what is homeostasis, maintaining the body temperature. Then nervous system, muscular system, circulatory system, respiratory system, excretory system, endocrine glands and their hormones, everything will be important. So the questions can be arise from the oxygen dissociation curve. If it is a right shift, what will be there? If it is a left excuse me, if it is a left shift, how the curve will be. Okay, so this is what an oxygen dissociation curve, the role of hemoglobin and nephron structure, hormone, that is gland matching, reflex are. So all these are all the micro topics that is very important. Say for an example, which hormones lowers the blood glucose? That is nothing but an insulin. These are all the basic questions. Okay, so the 14th unit will be this recombinant DNA technology. So in the recombinant DNA technology, first we are having a donor. Okay, so first one we are having a donor and we are having a recipient, and that recipient is nothing but a plasmid which is taking out from the E. coli. So we we have to break this plasmid with the help of this restricted endonuclease, and we have to ligate with the donor DNA with the help of this DNA ligase, and once again this recombinant plasmid will be entered into the E. coli, and each and for every 20 minutes this E. coli is going to reproduce. As a result of, we are going to get our target product. Okay, so this is what are all the enzymes which are involving recombinant DNA technology. What are all the vectors? Vectors here is nothing but a carrying vehicle. Here the carrying vehicle will be a plasmid. Genomic libraries. What is the difference between genomic library and cDNA library? cDNA means complementary DNA. Applications in medicine and research. So these are all very, very important. So that's all about the zoology syllabus analysis. Okay, so you based on the PYQ analysis, I have given you these questions. Okay, so if you follow all these rules and regulations to prepare before you have given an attempt to the exam, You keep in mind all these things. Okay, so how to approach each and every unit. Not all the units are similar. You have to approach differently for each and every unit. Okay, hope you find the session well. Thank you so much.